recombinant DNA technology, how to produce recombinant bacteria, kan? So all that we have looked during the first part of chapter 2. So for today's lecture, we are going to proceed to its application. Okay, its application. All right. Okay. Boleh dengar ya? Boleh nampak dan dengar suara saya?
Boleh, madam.
Okay, thank you. Okay, so examples of gene technology, which is in the production of insulin. So insulin is the hormones. Okay, hormones are classified into two based on its composition. So there are hormones that are made up of protein. There are also hormones that are made up of steroids. Okay, so insulin is a type of hormone that is made up of protein. Okay, so because it is made up of protein, so in order to synthesize the hormone insulin, the information is coded within within genes within the human genome. Okay, in the nucleus you have your DNA. So obviously inside your DNA you have genes that code for the synthesis of insulin. So the gene that code for the synthesis of insulin will be extracted. Okay, will be extracted from the human cell. Okay, because you can either, okay, it is you can extract out the gene for insulin by isolating the gene from, for example, pancreatic cells. Okay, because pancreas produces the hormone insulin. Okay, so you isolate the cell, you isolate the gene from the cell. Okay, from the cell, and then you cut. Okay, you cut the gene using restriction enzymes. Okay, using the restriction enzyme, and then you also isolate. You will have to isolate a plasmid from a bacteria. So usually the bacteria that is usually used for gene cloning. Okay, for gene cloning and also for the production of enzyme through bacteria is usually we used common bacteria is E. coli. Okay, so you need to extract, you need to isolate plasmid from the bacterial cell. Okay, so now you have isolated the plasmid from the bacterial cell, and the bacteria is left with no plasmid. Okay, so once you have isolated the plasmid, you also you put it in the test tube, and then you cut using the same restriction enzyme. Okay, you cut using the restriction enzyme as the restriction enzyme that you cut the human DNA. Okay, the human genome, to cut the gene of interest, which is the gene that is to code for the synthesis of insulin. So you cut because this will produce the same sticky ends. Okay, so when you produce the same sticky ends on the gene, and also same sticky ends on the plasmid, when the gene is inserted into the plasmid, the sticky ends will complement each other perfectly. Okay, so you after you have inserted the gene, you need to join. Okay, you need to join these two pieces of DNA together, which is the plasmid and also the foreign DNA, and then you need to join using the enzyme DNA ligase. Okay, so DNA ligase is to join. You can either use the word join or ligate. Sama je. Okay, insert the gene into the plasmid and join or ligate using DNA ligase. Okay, so this will seal the human gene and also plasmid together to form recombinant DNA. Okay, so after you have formed the recombinant DNA, then within that same test tube, you insert the bacteria that you have isolated its plasmid earlier, kan? Bakteria yang tak ada plasmid, awak masukkan ke dalam reaction tu. So reaction yang ada recombinant DNA dan juga bakteria yang tanpa plasmid. So they are there will be a host cell, which is bacterial cell that takes up the recombinant DNA. Okay, there are also other bacteria that does not take up the recombinant recombinant plasmid. So kita belajarkan hari tu macam mana nak screen. So how to screen is by depending on the dominant selectable marker. Okay, so the dominant selectable marker, which is ampicillin resistant gene. So when if the bacteria has taken up the plasmid, and if you grow the bacteria in a medium containing antibiotic, so the bacteria that has taken up the plasmid will survive because it has the ampicillin resistant gene. It causes the bacteria to be able to survive in a medium containing antibiotics. But if the bacteria that does not take up the plasmid, the bacteria will die. Okay, it will die off. And the second step of screening is the function of the reporter gene, which is the Z gene. Okay, so the function of the reporter gene is to screen. Screen is to select. Okay, to select bacteria that has taken up recombinant plasmid, and also bacteria that does not take up the recombinant plasmid. We call it as the non-recombinant plasmid. Bila awak cut open plasmid ni, kan, so ada plasmid yang dia akan ambil gene, kan, gene of interest, dan dia jadi recombinant DNA. Tapi ada juga plasmid yang mana dia tak ambil pun foreign DNA dan dia bercantum semula balik semula. Okay, so dia akan jadi non-recombinant plasmid. So kalau bakteria yang ambil non-recombinant plasmid, awak kena screen lah. You have to screen, you have to select only bacteria that take up the recombinant plasmid only. So that is the function of the reporter gene, which is the Z gene. So I have explained this in my video that I did to explain exercise on chapter 2 question 1. So if you dekat dalam video tu I have explained, okay, how to screen bacteria that has taken up recombinant plasmid from the non-recombinant plasmid. So I want you all to watch and understand the role, okay, the role of the Z gene, the reporter gene. Okay, so for the bacteria that has taken up the recombinant plasmid, okay, so now is we call it as the process by which, okay, the bacteria take up the plasmid, we call it as transformation. Ingat the process by which the bacteria take up the plasmid, okay, the recombinant plasmid, we call it as transformation. So now this bacteria, we call it as recombinant bacteria. Okay, it has been transformed, okay, into recombinant bacteria which has the plasmid that has the foreign DNA. So awak tahu ciri bakteria dia akan replicate banyak kali, kan, sebab it is bacteria, it will replicate very fast. Okay, so when the bacteria replicate very fast, it also copies the gene of interest. So that is the process of cloning. It makes many copies. Okay, it makes many copies of the gene of interest when the bacteria is dividing. Okay, so dia punya usage dia ada dua kat sini. Either you could isolate, okay, the gene of interest, which is the insulin gene, okay, would be cloned and then you extract out the gene, or you could isolate the product, which is the insulin, because the bacteria now has the gene of interest, which is the gene that can produce insulin. Now the bacteria can also produce the insulin. Okay, so you can isolate the insulin from the bacteria culture. Itu salah satu fungsi dia lah. Kalau kalau tengok yang saya bagi contoh hari tu, if you look at cheese products, kan, so ada the enzyme rennet. Okay, enzyme rennet can be synthesized, okay, by this method as well. Kalau tak synthesized by bacteria, it will be extracted from the intestine of calf, lembu. Okay, daripada dia akan ambil daripada perut anak lembu. Okay, so either. So those are the application of this technology. Okay. Okay, so now we are going to proceed with gene or DNA library. So a gene or DNA library is a collection of clone DNA. So kalau untuk DNA library, okay, so you have a large quantity of DNA in a human genome, for example. Okay, so you get the restriction enzyme to cut the genome of human DNA into DNA fragments. Okay, all DNA fragments ni you will insert into bacterial plasmid, and then you allow bacteria to take up the DNA fragment and allows the bacteria to replicate into many copies. So that is gene library lah kan. So it has all the information, all the DNA sequence of genome from certain species kan. So itu adalah dia punya definition dia. Definition. Saya tengok ada yang tanya soal yang ada yang tak faham dalam buku exercise workbook kan. Exercise workbook dia tanya what is gene library, and then dalam question MCQ kan. So yang pertama dia bagi adalah for DNA sequencing. For DNA sequencing tu purpose. Faham tak? Apa kegunaan dia? So berbeza dengan definition dia kan. Definition lain. Faham apa tu tudung? Tudung yang awak pakai ni. Kenapa awak pakai tudung sebab nak tutup aurat? Lain kan? Benda dia punya definition dengan dia punya kegunaan dia lain. Alright? So gene or DNA library. So it is a collection of clone DNA. So it has all the sequence of human genome. For example, kalau kita tengok genome library untuk human lah kan. So so the purpose is number one, collection and storage of genetic information of organisms. Okay, so we have gene library for different species. And then purpose for DNA sequencing. So you want to know the DNA sequence for gene for insulin. So what is the DNA sequence for certain gene? Okay, so after you have known the sequence, then you can synthesize. You can know what you can do further study lah kan, further study on the gene. So after you have known the DNA sequence, you can do gene cloning kan. So you make many copies of the gene and then you can insert into another organism, which is for example the bacteria, to allow the bacteria to produce the product, which is the protein. It can be enzyme or it can be hormones or other proteins. So DNA library is divided into two, which is genomic library and also complementary DNA library, cDNA library. C tu mesti kecil cDNA library. So for genomic library, it consists of DNA fragments representing the entire genome of an organism. Okay. So ini adalah sel. Dalam sel you have your DNA kan. So you isolate the DNA and then you cut using restriction enzyme kan. You cut using restriction enzyme, so you have many copies of your DNA. And then you insert into plasmid lah kan. So you insert into plasmid, and then plasmid will be taken up by the bacterial cell kan. So the bacteria will divide many times to form many copies of the DNA fragments. Okay. So it consists of DNA fragments representing the entire genome, keseluruhan DNA composition of an organism. Genome tu keseluruhan DNA of an organism. Okay, kalau kita ada 46 kromosom, semua sequence DNA sequence daripada 46 kromosom tu. Alright. So it contains all the gene from the 46 chromosomes. Alright. And then for complementary DNA, cDNA library, it contains only coding region of DNA fragments synthesized from mRNA. So for cDNA, it is synthesized from mRNA. Why do we synthesize the cDNA from mRNA? Because kita tengok kan proses the process of transcription. for eukaryotic cells, the process of transcription, after that it undergoes RNA processing, kan? So RNA processing will remove the non-coding sequence. It removes the intron, right? So after it removes the intron, the mRNA now has the continuous coding sequence. mRNA will have continuous coding sequence. Okay, coding sequence lah kat sini. So from this mRNA, you synthesize the cDNA, the complementary DNA. Complementary to what? To the template lah. Template now is the mRNA, dia terbalik. Kan, dia terbalik daripada process transcription. Transcription use the DNA as template to synthesize mRNA, but to synthesize cDNA, you use the mRNA to synthesize the cDNA. Okay, so now the DNA has the continuous coding sequence. For eukaryotic DNA, kita adalah eukaryot, kan? Eukaryot, kita punya DNA ni bukan semua adalah coding sequence. Dia antara coding sequence akan diselang-selikan dengan non-coding sequence. Okay, so in order for you to be able to clone DNA into plasmid, kan? So you need to produce DNA that has only continuous coding sequence. Okay. So 2.1.3, okay, so complementary DNA cloning and cDNA library. So DNA molecule made in vitro using mRNA as template for the first strand. So that is cDNA. DNA molecule synthesized in vitro in lab in test tube. Okay, it is synthesized using mRNA as template to synthesize the first strand of DNA. Okay, the first strand of the DNA, which is the first strand of cDNA. So cDNA clones have no introns. Okay, cDNA clones have no introns because it is synthesized by mRNA. Okay, mRNA as the template. mRNA has only exon. It is consists of only continuous coding sequence. So coding sequences might be interrupted by numerous introns. So this problem can be overcome by constructing clones of DNA copies of mature mRNAs from which introns have been removed. Okay. So the purpose is to produce DNA copies with only the coding sequence. Okay. So you need two types of enzyme, which is the first enzyme is reverse transcriptase. It is used to synthesize the single strand of cDNA using mRNA as template. Second, you will need DNA polymerase. So this DNA polymerase will synthesize the second strand of the cDNA and form the cDNA molecule, double-stranded cDNA molecule. So DNA polymerase akan menggunakan the first strand tu tadi, the first strand as template to synthesize the second strand. So cDNA clones can be expressed in bacterial cells because it does not have any intron. It does not have any non-coding sequence. Okay, so that's why you can clone cDNA into plasmid. Plasmid, you have to remember, it is double-stranded circular DNA of bacteria. So awak tak boleh nak masukkan mRNA dalam plasmid, kan? You cannot. So you have to produce double-stranded polynucleotide. In this case is the DNA molecule, is the cDNA molecule. Okay, then only you can insert the DNA into the plasmid to clone the gene. Okay, to clone the gene. Okay, so formation of cDNA clones via reverse transcriptase polymerase chain reaction. Okay, RT-PCR, polymerase chain reaction. Polymerase chain reaction is a technique to amplify many copies of the gene, okay, of DNA. So number one, formation of cDNA relies on RNA processing that occurs in the nucleus to yield mature mRNA. Okay, so this is your DNA molecule. Okay, DNA molecule in the nucleus. So DNA molecule in the nucleus, it consists of coding and non-coding sequence. It consists of exon and introns. Okay, so this intron is the non-coding sequence that is not useful to produce the protein. So during transcription, the first after transcription, it produces pre-mRNA. Okay, pre-mRNA. So this pre-mRNA, because it has the intron, it has to undergo RNA processing first to remove the intron to produce the mature mRNA that has only the coding sequence. It has only the exon. So now you have the mature mRNA. So mature mRNA is extracted and purified. Reverse transcriptase is added to tube to produce a single-stranded cDNA complementary to the mRNA. Dia macam ni, kalau awak nak synthesizekan cDNA, you must know what gene that you want. Kan? So maksudnya kalau awak nak gene untuk menghasilkan insulin, awak kena cari sel yang menghasilkan mRNA untuk hasilkan insulin. Hanya sel pada pankreas sahaja menghasilkan mRNA untuk insulin. Faham? Pankreas, sebab dia hasilkan insulin. Insulin is a protein. So in order to produce the insulin, is from the mRNA. Only the mRNA will be synthesized and expressed in the pancreatic cell. You cannot extract mRNA from the liver cell sebab liver tak menghasilkan insulin. Walaupun awak punya genome ni ada semua gene, kan? Dalam sel liver ada juga gene untuk insulin, tapi it is not activated. It is not activated, it cannot produce the mRNA for insulin. So you cannot extract mRNA from the liver cell, okay? To get the gene for to synthesize the cDNA for insulin, you have to extract mRNA from the cell that produces the protein only, okay? So isolate the mRNA for the cell that produces the protein, okay? So after you have isolated the mRNA, it is purified from other substance, okay, during the isolation process. So after you have isolated, so in this test tube, for example, you have the mRNA, okay. So you add reverse transcriptase into the reaction, okay. So reverse transcriptase is added to the tube to produce single-strand cDNA complementary to the mRNA. So the red strand here is the mRNA that acts as the template to synthesize the first strand of cDNA. Okay, so using the enzyme reverse transcriptase. So ini awak dapat combination between mRNA and also DNA. But because you want to synthesize DNA, you don't want the mRNA to be in your DNA molecule. So you have to degrade the mRNA using another enzyme. Okay, another enzyme, a specific enzyme will degrade the mRNA. So now you are left with the first strand of cDNA. So you add another enzyme which is DNA polymerase. So this DNA polymerase will synthesize the second strand of the DNA. Okay, the second strand of the DNA. So this produces the second strand and produces the complete cDNA molecule, the double-stranded cDNA molecule. Okay. So because this reaction occurs in a machine, we call it as polymerase chain reaction. So daripada satu molekul DNA ni, so melalui PCR polymerase chain reaction, the machine will amplify, will make many copies of the cDNA molecule. So now you have many copies of the cDNA molecule for the gene that synthesize insulin, for example. Okay, daripada satu copy, you have many copies for the gene that can synthesize insulin. This is just an example, okay? For example, insulin. So after you have produced the cDNA, so you can decide lah what to do with this cDNA. So you can either clone the DNA into the plasmid of bacteria, allows the bacteria to replicate and amplify further to produce the insulin, okay, that you can extract from the bacterial cell. Okay. So application of recombinant DNA technology. So recombinant DNA technology helps to examine fundamental questions about cells as well as new approaches to applied problems. Okay, so example, we can apply this DNA technology into investigation of certain criminal case. Okay, or you can produce GMO. GMO, genetically modified organism, transgenic organism, bukan transgender tapi transgenic organism. Okay, and then environment, can, so you could apply DNA technology to sustain the environment from pollution. For example, if you have contamination in the sea, can contamination of the sea by oil, can the sea is polluted by oil spillage, so you could produce bacteria that could uh, degrade away or eat away the oil spillage, okay, to clean up the ocean, for example. Okay, and then in the medicine field, okay, so kita tengok satu-satu its application in the medicine field. First, identification of genetic mutation associated with genetic diseases such as Huntington disease, hemophilia, and also sickle cell anemia. Okay, so you could investigate, okay, what happens to the gene, to your DNA that causes this different type of genetic disease. Kan? Second, for gene therapy, introduction of gene into an afflicted individual for therapeutic purposes. For example, the use of modified viral vectors to treat hemophilia B patients. So if you have watched my video for for exercise dalam buku awak tu, saya ada explain pasal hemophilia B ni, macam mana gene therapy tu berlaku. Gene therapy ni, it means that okay, kalau untuk hemophilia ni is a type of genetic disease where your blood cannot clot normally, secara normal. Sebab apa? Dia missing some clotting factors. Okay, so the person cannot produce certain clotting factors. So disebabkan apa? Certain gene are missing ataupun does not function well. So gene yang sepatutnya menghasilkan clotting factor dimasukkan ke dalam virus. It is inserted into virus. Virus as vector. Vector tu sebagai vehicle lah, kan, as a vector. So you insert the gene that can produce the protein, the protein. In this case is clotting factors. So you insert the gene into the virus, and then the virus will kita tujukan kepada organ yang bermasalah. In this case adalah pada, tak silap saya, pada liver, kan. So after the virus is inserted to the area of the liver, now the gene from the virus will be inserted to the liver cell, and now your body can produce the normal clotting factors, kan. So itu salah satu contoh gene therapy lah, kan. So you insert the gene to produce certain protein into virus. Virus dimasukkan ke dalam badan awak untuk menghasilkan protein yang sepatutnya badan awak hasilkan. Okay, to gene therapy. So the third example is the production of insulin for diabetic patient, macam tadilah, kan. So instead of kita isolate insulin daripada haiwan, kan, kita tak tahu status halal dia ke whatever, ataupun to get more insulin, kan. So it is synthesized through bacterial culture. Okay, and then B, genetically modified human growth hormone to overcome growth deficiency, kan. So you could also synthesize growth hormones, kan. And then and then the last one is produce vaccine for influenza A, hepatitis B, and also polio, kan. So through DNA, recombinant DNA technology is to produce vaccine as well. So virus yang digunakan ni bukannya virus yang boleh menyebabkan penyakit. Dia virus yang does not cause disease to human. Okay, ada nama dia. Right. Next, the second application is DNA fingerprinting. So it is for the analysis of DNA fragment extracted from individuals. Okay, analysis of DNA fragment extracted from individuals. So it is for analyzing evidence found at crime scene, identifying mass disaster victims, and also studying endangered species and clarifying disputed parentage and identifying human cancer cell line. Kejap. Saya nak kena
Ibu punya ni tak ada internet kan?
Saya tak suka dari animation ni. Sekejap. Okay. Okay, so these are the examples of the application in DNA fingerprinting. DNA fingerprinting ni, you analyze the a pattern of DNA fragments kan that you have run under PCR and also PCR and other methods kan, and then after that you do gel electrophoresis to get a pattern of DNA fragments kan. So this pattern can be used distracting to the animation ni. Okay. Okay, so DNA pattern ni kita panggil dia sebagai DNA fingerprinting. Okay, the pattern that you produced, okay, we call it as DNA fingerprinting. Bukan daripada ibu jari awak ke apa ke, dia lain, pattern of DNA fragments that you produced. Okay, through PCR and also gel electrophoresis. Okay. So macam ni, clarifying disputed parentage. So ada ada ibu ni, tak payah nak buat test, memang ibu tu yang keluar beranakkan anak dia, tapi yang the father kan. So the father, so mak perempuan tu accuse that guy as the father of her child, tapi laki tu dia tak nak mengaku. So kita boleh buat paternal test kat situ kan. So tengok dia punya DNA pattern dia sama ke tak. So that is one of the example. So and also to studying endangered species kan. So from there you could classify organism into new species ke tak kan. So if you found a new species, kita tengok dia punya genetic composition dia. So is it possible to classify that new organism as new organism ataupun daripada kita boleh masukkan dia pada spesies yang dah sedia ada, macam itulah. Okay. So identifying mass disaster victims kan. So those are examples of DNA fingerprinting. So almost all techniques for DNA fingerprinting rely on PCR amplification and gel electrophoresis. So saya dah mention tadi. Okay. So in order to get DNA fingerprinting, you have to isolate the DNA sample from cell, kan, from cell. Kalau contohnya ada crime scene, you get the blood stain, for example. So you extract out the DNA from that blood stain, ataupun from semen kalau ada case macam rogol ke apa ke kan. So you get the DNA sample. Tapi DNA sample yang awak dapat ni sangat sikit sebab you get that from a crime scene. So you have limited amount of DNA. So what to do is that you amplify the DNA. How to amplify is through the process of PCR, polymerase chain reaction. Polymerase chain reaction is to make many copies of the DNA sample. Okay, so after you have make many copies of the DNA sample, you insert into the reaction also enzyme. Can enzyme. So this enzyme will cut the DNA into DNA fragments. Okay, DNA fragments, and then you amplify the DNA, and then after you have make many copies, okay, you make many copies of the DNA, you run the DNA sample using this method, which is gel electrophoresis. Gel electrophoresis will separate the DNA fragments into its size. Okay, into its size. The larger fragments akan move slower kan during the gel electrophoresis. The smaller fragments will move faster. So this will produce DNA patterns yang kita panggil this sebagai DNA fingerprinting. Okay. So these are the two procedures yang kita buat. Okay, PCR amplification and also gel electrophoresis. And then second, a DNA profile of unknown suspect can be compared with millions of DNA profile in the database. Okay, so dia akan menghasilkan DNA profile lah kan, corak pattern DNA tu, kita namakan dia sebagai DNA profile. Okay, so kita boleh compare lah suspect punya DNA profile dengan dengan database, for example, ataupun father punya DNA profile dengan anak dia. So sama ke tak? So kalau sama, maksudnya tu adalah anak dia. Kalau tak sama, so bukanlah, contohnya. Okay. So DNA may come from blood, semen, kan, bones, teeth, hair, saliva, urine, or feces left at crime scene. Blood. Saya nak tanya, macam mana blood kita boleh extract DNA? Blood consists of red blood cells. Awak belajar kan red blood cells? Apa feature red blood cells? Does it have any DNA? Does it have any nucleus?
No.
So, macam mana?
White blood cell.
Daripada white blood cells kan. So white blood cells has the nucleus, okay, that contains the DNA. So nampak? So you have, because the sample is really little, and so you extract the DNA really small amount, so you have to amplify the DNA sample using PCR. Okay. Sekejap ya. Dia bagi situation kat saya pada surat khabar. Armed house robbery, 3 suspect arrested. So you have 3 suspect, kan? So you get the DNA sample from blood found at the crime scene. Okay, so all evidence have been sent to crime labs to identify the criminal. Okay, dramatic betul sehingga dia punya effect dia. Okay, so you have 3 suspect, kan? 3 suspect. So so you extract out the DNA sample. You run for PCR and also gel electrophoresis, and then you get the DNA profile. Okay, so from the 3 suspect juga you do the same thing. You get the DNA. You get the DNA from suspect 1. You get the DNA from suspect 2. You get the DNA from suspect 3. And then you for all the 3 DNA samples from these 3 individuals, from these 3 suspect, you do the same thing. You run for PCR, you run for gel electrophoresis to get the DNA profile for the 3 suspect. Okay. So this is the result. So ini adalah DNA profile for suspect 1, DNA profile for suspect 2, DNA profile for suspect 3. So this is what we call it as DNA profile. Yang satu-satu ni kita panggil sebagai DNA band, DNA band, 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 jalur DNA. Kan? So jalur DNA. So ini adalah DNA pada crime scene. So which suspect matches the crime scene?
Second.
Second. Okay, so ini adalah cara apa you get the DNA fingerprinting lah. Walaupun dipanggil fingerprinting bukan daripada cap jari tangan awak jari awak. Dia daripada DNA profile, the pattern of DNA fragment that you get after you do PCR and also gel electrophoresis. Okay. Okay, so next the third application is transgenic organism. Sekejap ya. Okay, so the third application is for transgenic organisms. So here you can produce GMO lah kan, genetically modified organism, transgenic plants for example, which is a genetically modified organism, GMO, to produce specific desirable and heritable traits. Ini kalau untuk contohnya tanaman pertanian kan, so agricultural crops for example, soybean, papaya, corn, whatever agricultural crops that you want to produce a better phenotype for that crop kan, so you could produce transgenic organism lah. Maksudnya kalau jumpa gene untuk menyebabkan papaya tu lebih resistant to virus infection kan, so you get the gene inserted into the cell of the plant to produce papaya that could resist virus infection. Okay, sama juga. So to produce corn with traits with better traits, such as for example resistant to drought. So you insert gene that allows the corn to be able to survive more during dry season, for example. Okay. So and then transgenic soybean resistant to polyphosphate herbicides. Soybean ni dia adalah sejenis pokok yang herbaceous kan. So bila awak tanam pokok soybean ni akan ada rumpai-rumpai juga yang biasalah bila awak tanam pokok akan ada rumpai yang awak tak nak, sebab rumpai-rumpai ni dia akan jadi sebagai pesaing lah untuk pokok ni dapatkan dia punya nutrients. So you apply herbicides. Tapi bila apply herbicides untuk soybeans ni, dia akan juga affected. Dia akan juga menyebabkan soybean ni mati. So you insert gene in that soybean so that the soybean is more resistant to the herbicides. Awak hanya akan kills the kita panggil tu lah, rumpai itulah kat sebelah dekat sekeliling dia. Okay. So those are examples of transgenic plants. Okay, next is for environment. So transgenic microorganisms like Pseudomonas, Pseudomonas bacteria, to break down hydrocarbons in oil and clean up oil spillage. So you produce bacteria that could eat away oil spillage in the ocean. Okay. So this is the Pseudomonas that could eat the oil spillage in the ocean. It breaks down. Okay, it breaks down the hydrocarbons in the oil. And then transgenic microorganism that can extract heavy metal in environments such as copper into copper sulfate or lead sulfate. Okay, for bioremediation, so using biological organism to clean polluted areas. Okay, so such as for example Saccharomyces cerevisiae, okay, which is common baker's yeast. So ini untuk hasilkan yeast lah. Okay, so dalam awak punya nota ni dia ada the last two subtopic which is gel electrophoresis, and then The last one is PCR. Okay. So selalunya kita akan buat PCR dulu, kan? Kita akan buat PCR dulu baru kita buat gel electrophoresis. So saya nak explain PCR dulu. Okay, I want to explain to you the PCR part. We have to amplify the DNA first. Okay, make many copies of the DNA segment, the gene. Then only you can run for gel electrophoresis. Okay. So what is PCR? So PCR is a method to amplify or make many copies of specific DNA segments in vitro. In vitro, it means in the lab, okay? Or another term is replication of DNA in a test tube. So you prepare your DNA sample, you insert your DNA fragments, your other reaction substance dalam tu kan. You put the NTPs, you put enzymes into that test tube. Okay, test tube ni bukan test tube yang macam dalam punya lab besar yang letak all the chemicals. Yang kaca tu tak, dia it is a small test, it is a small tube, small je, really small. To insert your reaction, okay, your DNA, your NTPs, your enzymes, okay, into the reaction. Small, just a small tube. Dia bentuk tiga macam ni lah, macam tu je. So dia ada penutup untuk tutupkan dia macam tu. So this, kat sini lah letak awak punya DNA sample. You insert your enzymes, polymerase, the NTPs adalah monomer to synthesize, monomer of nucleotides to synthesize the polynucleotides. Okay. So tools required in PCR is the thermal cycler PCR or DNA amplifier machine. Okay, PCR ni sebenarnya dia adalah sejenis mesin. Dia menggunakan sejenis mesin yang macam besar, kecil, lebih kurang kecil sikit pada mesin, daripada printer. Kan? The machine is something like this. Okay, so nampak? So this is where you will put your tubes containing your DNA sample. Kecil je kat sini. So here is the setting lah. You put your setting, berapa temperature yang nak letak, berapa cycle kan. So you boleh juga simpan memory for your setting tu. Sebab you have many sample to run kan. So setiap sample tu berbeza setting dia. So you could store the information in this machine as well. Okay, for the PCR for different DNA sample. Okay, so next. So another tools that you need is free nucleotides. We call it as DNTPs. Okay, DNTPs adalah the monomers of nucleotide that you require to synthesize or copy the DNA molecule. And then your DNA sample, and obviously you have to have your DNA sample daripada crime scene ke, for example, DNA sample from certain individual to do certain tests, kan. And then PCR primers, kita dah belajar DNA replication kan. So DNA replication in order for DNA to be replicated, it has to have primer kan. It has to have primers. And then the enzyme tag polymerase. Dalam DNA replication yang kita tengok dulu, untuk to synthesize the new strand, it requires DNA polymerase. But DNA polymerase cannot withstand high temperature because when you run for PCR, dia melibatkan suhu yang tinggi kan, to denature to split the DNA fragments kan. Then the temperature involved is really high. So you cannot use the normal DNA polymerase. You have to use this enzyme which is tag polymerase. Tag polymerase is an enzyme that you extract from bacteria that lives in hot spring. So it can withstand high temperature. It will not be denatured during the process, during PCR. Okay, so primers. Kita tengok the first one dulu, the PCR primers. PCR primers are short single-stranded DNA sequence of around 20 nucleotides long. Okay, so 2 primers are used in PCR for each, one for each. So this is our DNA sample. Okay, our DNA sample, DNA is double-stranded. So the DNA molecule, the double-stranded DNA molecule is held by what? Hydrogen bonds kan, between the bases. So during during PCR, so these hydrogen bonds has to be broken. It has to be broken. So you produce DNA templates, two DNA templates here. So when you produce two DNA templates, obviously each of the template have to have their own primer. Okay, so the primer will start the process of replicating the DNA molecule, the original DNA molecule. Okay, so PCR primers. So of about, it is a short sequence of 20 nucleotide base pair. Okay, base pair, BASE pair, base pair. Okay, so the next tools that you require is tag polymerase. So PCR requires DNA polymerase to synthesize new DNA strand. So tag polymerase was isolated from this bacteria, which is Thermus aquaticus, that lives in hot spring. Okay, so it can withstand high temperature of about of up to 95 degrees Celsius. Okay, so when you run for PCR, obviously to break the hydrogen bonds, the temperature is really high, and also to attach all the nucleotides, it also requires high temperature of about 70 degrees, something like that. So this temperature could denature the normal DNA polymerase. So you need to use tag polymerase because it can withstand high temperature. It is heat stable. Yang ini, point ni pernah keluar dalam soalan exam batch lepas ke tak silap saya, kan? So this is the picture of tag polymerase. Okay, why do we use tag polymerase? Okay, so the cycle 1, produce two identical DNA molecule. Okay, so the first step is denaturation. So original samples of target DNA exposed to high temperature. Okay, so high temperature is to what? Is to break the hydrogen bonds here. Okay, to break the hydrogen bonds between the DNA molecule. Okay, so that it can become DNA templates. Okay, so that is denaturation. So increase temperature to break hydrogen bonds between the two DNA strands. Okay, so denaturation can reach up to 94 to 98 degrees Celsius. So next is annealing. So annealing is what? DNA fragment is then cooled to allow primers to bind to each strand. So after you have separated the DNA template, DNA molecule, it becomes the template. So in order to start the reaction, you have to have primers. Okay, PCR primers. So these are the primers that will be that will bind to the DNA template during annealing. So this is the second stage of PCR. The third stage, after you have attached the primers, is extension. Okay, it's extension. Okay, so extension requires free DNA nucleotides that are complementary with the original strands are added by tag polymerase and rise the temperature to form new strands of DNA produced to identical DNA molecule. So you have attached your, so you have your DNA template and separated during denaturation. So during annealing, you attach the primers, PCR primers, and then extension, the temperature is about 72 degrees Celsius. So the tag polymerase will add free nucleotide, which is the DNTP study, DNTPs, according to the template sequence. So it must complement. Tag polymerase must add DNA nucleotides complementary to the DNA template. Okay. Sekejap. Okay, so these are the three. PCR ni dia ada banyak cycle. This is the first cycle. For second, third and subsequent cycle, dia akan repeat proses yang sama. Denaturation, annealing, extension. Okay. Okay, so after 3 cycles, 2 molecules match the exact sequence exactly. Okay. to match the target sequence exactly. So after 30 more cycles, over 1 billion, okay, molecules match the target sequence. So this is the original target sequence, okay, which is DNA fragment from the whole genome. So how to get the DNA fragment? You cut using specific enzyme. You cut using specific enzyme, and then you have the DNA fragments, and then you amplify the DNA fragment through PCR. So PCR, for the first cycle, it is denaturation, and then annealing, and then extension. Denaturation is to break hydrogen bonds between the two DNA strands so that it becomes the template. So during annealing is to attach the primers. Okay. So annealing cools to allow primers to form hydrogen bonds with the ends of the target sequence. And then extension ataupun elongation, where DNA polymerase to be exact is the tag polymerase, and DNA nucleotides to the 3' end of the primer. Okay, so sama je application dia. Every time that you need to add DNA polymerase is always at the 3' end. Okay, so after 2 cycles it yields 4 molecules. Okay, 4 molecules after 8 cycle. After cycle 3 it produces 8 molecules of DNA. Okay. Okay, so kat sini yang dalam punya exercise tu kan, so dia bagi berapa cycle, you have to determine how many DNA molecules are produced. So formula yang you have to use is 2n. Okay, 2n is the number of cycle, then you get how many DNA molecules produced for that cycle. Okay, so kalau 30 cycle, you just put 2 to the power of 30, you get how many DNA fragments that you produced. Okay, that the PCR will produce. Sekejap. Saya try to play this video. I will try to play this. Saya nak save dia kejap Save media. Okay, so I will play the video. Okay, so hopefully you can hear the video well.
The polymerase chain reaction is a method for making many copies of a specific segment of DNA, starting with a very small amount. This technique can be used to identify specific microorganisms from a small amount of DNA and to identify persons involved in crimes from DNA on cigarettes or in a single hair follicle. The DNA to be amplified is mixed with deoxyribonucleotides, a thermal stable DNA polymerase called Taq polymerase, and DNA primers. The DNA primers hybridize to the ends of the gene to be amplified and provide a starting point for the Taq polymerase. The mixture is heated to break the hydrogen bonds in the DNA, forming single-stranded molecules. The mixture is then cooled sufficiently to allow the DNA primers to anneal to each end of the segment to be copied. Taq polymerase then synthesizes the complementary strand of DNA using the primer as the starting point. The temperature is raised again to separate the DNA strands and then lowered sufficiently to allow the primers to attach. Taq polymerase now synthesizes another set of new complementary strands. This process is repeated until enough DNA has been produced to be identified or used for further research. After 21 cycles, one molecule of DNA can be amplified to over 1 million copies. This amount of amplification can be achieved by running the reaction overnight in a thermal cycler, an instrument that automatically raises and lowers the temperature at appropriate time intervals.
Okay. Okay, so far, do you have any questions? Habis dah chapter 2 ya? Any question before we proceed to chapter 3?